Okay, well, it is 7 o'clock. Shall we start this right on time, Waylena? Why not? All right. Well, folks, thank you all so much for joining us. I'm going to switch my view so that you folks see me talking here for just a moment. We thank you so much for attending the James B. Kaler Science Lecture Series, so much so that, you know, we doll ourselves up for it. That's why I'm actually wearing a suit tonight. Not a common occurrence for me. But nevertheless, we have recruited some wonderful speakers for these, and this is the first of our talks for the year 2021. And our speaker today is Dr. Jessica Conroy. She's a professor in 2 departments at the University of Illinois, in both the Department of Geology and the Department of Plant Biology. And she earned her BA in geology from the College of Worcester and her MS and PhD from the University of Arizona. She has been working for the University of Illinois since 2013, but beforehand she was a postdoctoral fellow in the School of Earth and Atmospheric Sciences at Georgia Tech, which was funded by the National Science Foundation. Now, I will just share with you folks, in case you're curious about how the name of the island that is in the Pacific Ocean is going to be the focus of today's talk. Just to spoil everything Jessica was going to say, it's usually pronounced Christmas in the Gilbertese language. That those consonants are pronounced in that way. It's just something I've always had fun with. It's what, Kiribati is the name of the country, is that correct? She's nodding, so maybe that's my cue to stop talking. So anyways, if you are an attendee that's here to earn some extra credit from seeing this great talk, I would ask you kindly to rename yourselves so that the name of your instructor is listed among your names. You all have the ability to rename yourselves. And even though your instructors can't see who's here, I will be able to send reports out to them at the end of this talk about who attended, all right? So without any further ado, Dr. Conroy, the floor is yours. Thank you so much.
Just a moment. Okay, how are we seeing everything, folks? Looking good? Thumbs up? Fantastic. All right, I'm going to get going here. Well, thank you so much for joining in on a Friday evening to hear about my research. I hope you learned something new and interesting. And so I'm going to be talking about paleoclimate and geobiology and information about these very broad subjects that we can glean from lake sediments on the tropical Pacific island called Christmas Island. All right. And, you know, before I get into the science, maybe you just want to know a little bit about who you're going to be listening to for the next 45 minutes. So Eric already gave a very nice introduction, but I'll just add to that, you know, my work very broadly is focused on understanding how the climate system operates. And that involves both, you know, in the past and in the present with an eye towards the future, with the goal of, you know, improving our understanding of future climate change with increased greenhouse gas emissions. So a lot of what I do is paleoclimate data collection, which we'll be hearing about today from lake sediments. I also work on aeolian sediments, which are wind-blown sediments. And I also do quite a bit of work with coral paleoclimate data. I also spend quite a bit of time these days looking at climate model output and comparing climate models and paleoclimate data to improve our understanding of past climate dynamics. And I have a special interest in the water cycle. So in particular, I use something called stable isotopes of water to understand how water moves the water cycle. And I'll tell you a little bit about that today. And the goal there is, again, going back to the past. This helps us improve paleoclimate interpretations of past water cycle variability. And here's my group. This is before COVID, when we were all together in front of our home, which is the Natural History Building. And, you know, science never happens in a vacuum these days, right? So I want to acknowledge everyone who's made today's talk possible. And first and foremost, that's my students. And so I really have the privilege of telling you about all of my students' great work in the Conroy Lab. And of course, I have many wonderful collaborators, field assistants, and I've had some great funding sources that's paid for all of this work to happen. Okay. All right, well, this is the outline for today's talk. So I'm going to start with more of a lengthy intro telling you about, where we're going today and why I do what I do. And then I have sort of 3 different scientific vignettes for you. Okay, so if I do lose you at some point, and I know that happens these days with our attention spans, know that you can always jump back in at the end of each of these, 3, or well, I guess 2 scientific vignettes and the introduction. Okay, so I want you to imagine that you're in the International Airport Terminal in Honolulu, and you're about to get on a plane. Kind of hard to imagine, right? And you're going to be whisked away to Christmas Island. And so where is Christmas Island and where are you going? So Christmas Island is one of the islands in the Republic of Kiribati, as Eric mentioned. It's actually the world's largest coral atoll by surface area. And this is what it looks like on Google Earth if you were to zoom in. And you can see it's got sort of this claw shape. Here's the lagoon, the main lagoon up here in the northwestern quadrant. And you see there are hundreds and hundreds of lakes on this island, which is part of the reason I work there. And just to sort of orient you geographically, Christmas Island is one of the northern line islands, okay? And so these are about, if you're on that plane, it's about a 3-hour plane ride south of Hawaii. And we're sitting on Christmas Island at about 2 degrees north of the equator. There are a few other line islands as well. Palmyra Atoll is actually United States territory, and there's a research station there. And there are a few other smaller islands. Later on in this talk, I'm going to compare a paleoclimate record from Christmas Island with a paleoclimate record from this island, which is also called Washington Island in the literature. So just remember that. So maybe some of you have heard of Christmas Island before, and one reason you may have heard of it is that it has a bit of a dark history, and that it was the site of above ground nuclear testing in the 1950s, primarily by the UK. And so unfortunately, a lot of atomic bombs were dropped in the atmosphere, detonated in the atmosphere above Christmas Island. And, this had devastating effects on the health of the servicemen stationed on the island, as well as the villagers who were exposed to this radiation. So, that's a sad history of Christmas Island. And there's still kind of remnants of that military history you can find across the island in terms of like an old air base and things like that. But why, as a paleoclimatologist, someone who's interested in the Earth's climate system, why would I want to go to this remote location to study climate change? Well, Christmas Island is really in a sweet spot. It's in a place that we're going to call the ENSO Center of Action. So what's ENSO? It's the El Nino Southern Oscillation. It's this 2 to 7 year, or what we call interannual oscillation that involves both the ocean and the atmosphere in the tropical Pacific. Okay, and so we have El Nino events and La Nina events, which maybe you've heard about in the news. And I want to describe what these events are to you. Understanding El Nino and La Nina are going to give you a really good sense for how the tropical Pacific climate can behave on a variety of timescales. So it's a really good primer in that sense. Okay, so sort of normally, and La Nina is kind of an enhancement of normal, during these times we have strong trade winds that blow on the surface from east to west across the tropical Pacific basin. So they converge on the equator and they blow from east to west, okay? And what that does is it does a really good job of pushing the surface water in the ocean over towards the west where it piles up. And then cooler subsurface water rises from the subsurface of the ocean, from the deeper parts of the ocean, to replace that surface water. Okay, so ocean temperatures here are quite a bit cooler than you would normally expect on the equator. Over here in the Western Pacific, though, you've got warmer ocean temperatures. You also have warmer air temperatures, and you have this process of convection, warm air rising because it's less dense. And that warm air also holds a lot of water vapor. And so you get a lot of precipitation over this warm body of water in the Western Pacific. Okay, and this area of rising air and precipitation is one branch of a very important circulation cell we call the Walker circulation cell. So you have warm air rising over here. And once that air reaches the top of the tropopause, it kind of diverges and it's stripped of its moisture by precipitation processes. And then cooler, drier air from high up in the atmosphere subsides in the Central and Eastern Pacific. So over here in some, then it's cool and dry. And over here, it's warm and wet. And then the star there is where Christmas Island is located. Now, during an El Nino event, this all switches around. So maybe those trade winds weaken. Maybe there's an anomalous burst of westerly winds coming from the Indian Ocean. And what happens is this warm water sloshes back into the eastern and central Pacific, and rainfall follows those warmer waters. So it becomes warm and wet where it's normally cool and dry, and it becomes drier where it's usually quite wet. Okay, so everything switches around during an El Nino event. So this has big consequences, obviously, for the climate of the tropical Pacific, right, and the weather of the tropical Pacific. But why we care so much about what's going on in the tropical Pacific is that unlike the Las Vegas model, what happens in the tropical Pacific doesn't stay in the tropical Pacific. So these changes ripple across the world in the form of what we call atmospheric teleconnections. And this word teleconnection, what it means is when you have a climate event or a change in one place that influences climate and weather farther afield in a very remote location. And so ENSO winds up influencing climate around the world. And this is why we want to understand the tropical Pacific climate system. And that holds for ENSO, that 2 to 7 year oscillation that we've been talking about, and also for how the tropical Pacific behaves on longer timescales as well. Okay, and so just to give you a taste of that, so the tropical Pacific can also vary on decadal timescales. And so a decadal means, every 10 years or so. And so this is a really cool plot that it really illustrates that. And so what you're looking at in black are temperature observations, annual mean for the globe, okay? And so you see that steady march upward, right? We know that's happening. The world's getting warmer. But superimposed on top of that are lots of ups and downs. And what drives those ups and downs? Well, it turns out from this modeling study that the model couldn't simulate reality unless it cooled down the Eastern Pacific. Okay, so what's going on in the tropical Pacific Ocean influences the strength of the global warming trend as well. Okay. All right. So since the tropical Pacific is so important on these timescales, it's really critical that we're able to model the tropical Pacific climate system so we can project how future climate is going to change. Okay, it's the critical part of climate models. But unfortunately, of all the climate models we rely on to project future climate change, they just can't get ENSO right. There's just a lot of trouble simulating the tropical Pacific climate system. And so there's a lot of uncertainty about what ENSO is going to do in the tropic and just tropical Pacific climate in general on those longer timescales going up towards 2100, in 80 years. And so that's a problem given how much what's going on here can influence, even how much the world is warming. So why is it such a problem to simulate the tropical Pacific climate system? Well, climate models are validated or tested with modern observations, modern climate observations of temperature, precipitation, atmospheric pressure. And so what's going on in the tropical Pacific? Well, there's not a whole lot of measurements. And so what I'm going to show you here is just kind of snapshots of different months from 1880 through the mid 20th century. And what you're looking at in the colors are just the number of times ocean temperature or sea surface temperature, SST, was measured in those 1 degree by 1 degree squares. So that means a ship was coming by, they dropped a bucket in the ocean, pulled it up, stuck a thermometer in it, and recorded that temperature. And the take home point for you here is that, gosh, there's a lot of white space, right? We don't have a really good constraint on temperature in this really important region, even going into the mid 20th century. Okay, and so we have a really limited record to ground truth those models. And there are various sophisticated techniques we can use to infill these empty spaces, so that does exist. But at its heart, we just don't have a lot of data from this region. And so this is where my field of expertise comes into play. So we can extend the instrumental record back in time in this very important location using paleoclimate proxy data. And when we do that, we can get long paleoclimate records, and we can look at how things like ENSO changed on long timescales through centuries and millennia. And we can look at how sort of what we call, background or mean state changes have occurred through centuries to millennia. Okay, and so that whole field is called paleoclimatology. It's the study of Earth's climate prior to the period of instrumental observations, which started, I'd say, around the beginning of the 20th century. And even in that case, in the tropical Pacific, as you were able to see, you don't have a lot of data. So how do we do that? We rely on something called natural climate archives. So archives are on the landscape or in the ocean that somehow are recording the environment in which they grew or accumulated. And so there are many different types of paleoclimate archives. They include things like tree rings, like coral skeletons, ice cores, cave deposits, and my own area of expertise, sediments. We can obtain sediment cores from lakes and from the ocean. And those are records of sediment that's been accumulating slowly, maybe a millimeter a year in lakes over thousands of years. And we can measure lots of things in those cores to tell us about how the environment and how the climate has changed through time. Okay, so that's what we're going to be talking about today. And we're going to focus in on one particular time period in this talk, and that's the last millennium, or, roughly the last 1,000 years. Although we like to tack on a few decades to, prior to 1000 CE and start at 800. So why do we care about this time period? Well, we can think of it as kind of a laboratory to investigate how the Earth's climate responds to when it's poked by natural forcing factors like volcanic eruptions or changes in solar radiation. And we can also investigate how the Earth's climate just varies internally in the absence of what we call those types of forcing. And this provides a really excellent baseline that we can use to assess the anomalous 20th century when we started pumping greenhouse gases into the atmosphere and warming things up and changing things dramatically. And so during this time period, which you can see here, and this is this sort of gray to white shaded area, this is a compilation of many different independent temperature reconstructions of this time period. And so where it's gray, that means all those records agree really well on what the temperature was. And where it gets lighter towards white, there's divergence. So it's sort of the envelope of uncertainty that we have. Okay. In this time period, we have these two chunks of time that people like to focus on. There's this time period called the medieval climate anomaly. And this is a period where some parts of the Earth were quite warm, but not the entire Earth. So it wasn't warmer everywhere on Earth. So that's really important. So unlike the 20th century where, I'd say most places are warming to some degree, during the medieval climate anomaly, this seems to be focused in Europe and in parts of the Northern Hemisphere. Okay, but it's still a pretty big signal. And then we have the Little Ice Age, a cooler period, but again, not globally uniform, but still pretty important. So a big question is, what was the tropical Pacific doing during these time periods? And why do we care about that? Because whatever the tropical Pacific was doing, that could wind up influencing things like whether it was warmer or cooler than average or drier or wetter than average in many remote places where we have those teleconnections. Okay, so what are some of the hypotheses that are out there for what the tropical Pacific was doing during the last millennium? So this is kind of where things get fun because there's not a lot of agreement. And this is why we get funded to do our work. So one of these hypotheses is that there is this belt of rain that straddles the equator of the Earth, or generally just north of the actual equator on what we call the thermal equator. And we call that the intertropical convergence zone. Okay, so air is converging, it's warm, it's rising, we're getting convection, lots of precipitation. Okay, this is the rain belt of the Earth. So one hypothesis suggests that over the tropical Pacific, this rain belt shifted southward during that time period, the Little Ice Age. So there are some records that support that. And that's going to change, where it was wet and where it was dry across a very large region. Another hypothesis is maybe, this rain band, maybe it didn't just migrate, maybe it just got wider during the medieval climate anomaly, maybe it contracted during the Little Ice Age. Again, there's some records that suggest that as well. And then a final hypothesis is thinking about the tropical Pacific more in terms of that circulation cell I discussed earlier when I was explaining El Nino and La Nina, that Walker circulation cell. That is a feature of the climate system of the tropical Pacific that's always around. It changes during El Nino and La Nina events, but it can also get stronger or weaker on longer timescales. And so this hypothesis suggests that during the medieval climate anomaly, this Walker circulation was generally stronger, which means you'd have really wet conditions over here and dry conditions over here and in the Central Pacific. Okay, so, these hypotheses are competing, so there's more work to be done. And so what I would argue is that the records that we're developing from Christmas Island are really a critical puzzle piece to test these hypotheses. And I want to illustrate that, especially in comparison to a pre-existing record from nearby Washington Island, which is just north of Christmas Island. Okay, so today, Washington Island is a very wet place. There's a lake on Washington Island, which has been cored, and the climate record develops from that sediment. And so that's what we're going to be comparing to our Christmas Island record. So today, Washington Island is very wet. It sits under this rain belt. This is mean ITCC precipitation. So you can see there's a lot of rain in this band right here. Christmas Island, though, is quite dry. Christmas Island is always south of the ITCZ. So it's very arid, except when there's an El Nino event, and it gets very, very wet on Christmas Island, as well as Washington Island. So if there was a change in the position of the ITCZ, Washington Island should get drier while Christmas Island gets wetter if there was a migration. If there was a change in the Walker cell, both sites should get wetter or drier. Okay, so we can start to test these hypotheses with this new record. Okay, so that's the scientific motivation for why I'm doing what I'm doing. But I wanted to just share a little bit of kind of the human story of how I got to work on Christmas Island to begin with, because not many people work here scientifically. And so as a postdoc, I worked with a coral paleoclimatologist who would go to these line islands and collect fossil coral cobbles, as well as she would also drill modern corals. And she can use those records, those carbonate records, to reconstruct changes in the ocean, in particular, El Nino Southern Oscillation variability through time. So these are the best records we have of ENSO variability. Okay, and so I went there with some pretty great people and spent 2 weeks collecting fossil corals, labeling them, and we'd ship big bins of them back to the States to be analyzed in the lab. But, you know, as I was there working in this amazing environment, I had already been trained as someone who studies lakes, a paleolimnologist, someone who studies lake sediments. And so I couldn't help but see all of those lakes on the landscape. And boy, they were fascinating lakes. I'd never seen anything quite like it. Many of these lakes are very saline, so they're what we call hypersaline. And so they contain these microbial mats, which we'll talk about later. And it's really just a stunning and kind of different and almost alien kind of landscape to work in. And so I immediately wondered, can these lakes hold a record of past climate change? And can we use those records to complement coral records? The corals are doing a really good job of reconstructing ENSO because we can measure monthly changes in the coral skeletons. Lakes, you can't really measure monthly changes in the sediment record, but you can get these really long continuous record of changes over, you know, decades to centuries, which is also very important. And so I went with my first PhD student, Melinda, who is now a professor herself, as well as Brandon Curry from the ISGS, who's a coring, extraordinary coring expert. And we obtained some sediment cores with a little bit of seed funding from UIUC. And so now I'm going to start digging into the science, and I'm going to tell you about those sediments. But first, I want to take a step back and talk to you about these lakes in general. And one of the questions we had was, how do these lakes respond to climate variability in the first place? And so, you know, this question is motivated by a need to understand the modern system if we're going to accurately interpret climate from the, you know, the remains of the ancient system, the lake sediments. Okay, and so what my student Melinda did is she used LANDSAT imagery to look at changes in surface water area across this island through time. And so she developed an algorithm to create a water to land ratio for the exposed land on Christmas Island, you know, excluding tidal influences and things like that. And so the gray pixels here are surface water and the black are land. And, you know, of course, clouds were an issue because there's always a cloud somewhere on this island. There's only like 6 instances where we don't have a cloud. So she resolved that by looking at sub-regions of the island. Okay, and this was all motivated because we were playing around on Google Earth before going in the field, and we noticed that these lakes change a lot in size through time. And we wanted to know why. And, you know, the big take home is pretty straightforward and intuitive, that when you have these intense periods of precipitation during El Nino events, there's a lot of rain, a lot of runoff, a lot of groundwater flow, and these lakes expand. And then when there's not an El Nino event, they slowly evaporate. And so surface water area shrinks. Okay, and you can see that hopefully pretty clearly in these comparison images from the same locations during a dry period and then a wet period. So you can see the gray pixels kind of infill all this black area. And so this is the lake expanding during a wet period. And most notably, we saw that there's this El Nino event that happened in 2015 to 2016. It was really big. We call it the, the monster El Nino event. And this is probably the strongest event recorded in instrumental history. So, and that's, starting in the late 19th century. So it was a big one. And so that event caused an extremely large deviation in surface water area. So a big increase in surface water area. And this has societal implications as well. So, when these lakes expand, they flood the roadways on Christmas Island. So the islanders can't drive to various villages because the roads are flooded. It's also a breeding ground for a big mosquito population. And so we have, anecdotal evidence that there is an uptick in mosquito-borne illnesses during these times, these wet times. And, my colleagues I think one of them got dengue fever during this time when they were down on the island. The other got chikungunya. So there are implications to the surface water. So apart from that remote sensing study, we're also doing a lot of modern monitoring of these lakes to try to understand these lake systems. And so that involves detailed hydrologic monitoring of lakes where we're actually coring the lakes and obtaining the sediment records and doing kind of more broad surface water measurements and sediment collections from a lot of different lakes. This is one of my current PhD students, Allie Wyman, augering down, digging a hole for a well that she's about to install. And Allie's about to defend her dissertation in a couple weeks. So Allie, in that previous picture, was working at a site called Lake 30. So all of the lakes on this island, there are so many of them, they're just numbered. They don't have names. So this is Lake 30. Okay, and so Lake 30 is a kind of a special lake in that it's a groundwater fed lake. So it's not a lake that's connected to this main lagoon and fed by seawater. It's fed by groundwater. And this is what it looks like. Okay, so it's very shallow. Most of the lakes on this island are. And you can see there's really there's not a lot of topography. It's very flat here and it's very small. Okay, and there's no surface water, no stream draining in or draining out. It's what we would call a closed basin from the surface. However, with all of Allie's hydrologic monitoring, what she's uncovered is that inflow and outflow in the subsurface changes considerably from field season to field season. So in one field season, this lake was a bit smaller and it was also a bit saltier. And she noticed that water was flowing into the lake, but not flowing out of the lake as much, except for into this little pothole here that's nearby. Then in another field season, the lake was noticeably bigger and water was flowing into the lake and also out of the lake. Okay, so there was subsurface outflow. And the salinity, when we measured the lake this time, it was basically fresh. And so that salinity speaks to the water residence time. So when water is sitting around in this basin for a long time, it's going to evaporate and that increases salinity. Okay, if it's going in and then going out, it's going to stay fresher. So overall, what this means is that, salinity and inflow and outflow are really tightly connected with what's going on in the atmosphere with, how much it's raining versus, evaporation rates. And so one interesting thing is that we noticed an enormous salinity change in many of the lakes on Christmas Island between our field seasons. And so what we think this is that massive El Nino event caused these lakes to freshen quite a bit. And we're hoping to look into those, comparing those data sets from before and after that big El Nino in another study in the future. Okay, but let's get into the lake sediment finally. So these are what the cores look like that we're taking in the field, okay? So they're tubes of sediment and we bring them back to the lab, cut them open, and we can measure a whole bunch of geochemical, of biological, of physical properties of these sediments, okay? And so here we are during one of our field seasons. This is pre-COVID. The reason our faces are covered is that we're coming from central Illinois and the sun at 2 degrees north is intense and it will basically burn your skin off if it's not covered. So we're covering our faces so we don't get massive sunburn. We're also coated in lots of sunscreen, of course. It just actually physically hurts. But we're kind of goofing her off on this picture too, having a really good time in the field. Okay, so when you have one of these cores, the first thing you need to know is how old it is, right? If you're going to reconstruct climate change through time, how old is this sediment? And so we rely on radiocarbon measurements to create what we call an age model. And so this is the age model for Lake 30. And so we have depth here and calendar years before present here. Okay, and so, we have the youngest sediment near the surface, the oldest sediment deepest in the core. And we see a really nice age model here. Sediment, it indicates sediment was slowly and continuously accumulating through time. And then suddenly there's a big break. And what that means is that there was a hiatus. So either, we think the most parsimonious explanation is that the lake dried up. And so there was no sediment deposition. And so that's what the hiatus is. And then we get the lake merging again and depositing sediment in the last, I'd say, 4 decades. This hiatus occurs during a little ice age. So this is actually a critical and interesting piece of information, I think. So what else did we measure in this core? Well, you can measure a lot of things, and I could keep you here all night, but I'm just going to talk about 2 of the things we measured today. So the first and most important thing we measured are the stable isotope ratios of hydrogen that are preserved in lipids that are found throughout this sediment core, okay? And we measured this because there is an existing quantitative relationship that lets us turn that measurement value into salinity, okay? And so this delta value, this might look very stressful. So just to make it not stressful, what we're looking at are stable isotopes. So just a quick primer, if you don't remember, an isotope is a variation of an element with a different number of neutrons than another variation of the same element. So a stable isotope is one that's basically not radioactive. So these aren't decaying through time. They just stick around as they are. And so in the case of hydrogen, we're looking at a hydrogen with an extra neutron versus your standard hydrogen, okay? And so that delta is the notation that we report those isotope ratios in. So for delta, and it's usually paired with oxygen 18, so that's why this is on the slide. We're looking at the ratio of those hydrogen atoms in a sample relative to a standard, which in this case is something called Vienna standard mean ocean water, and then dividing by the standard and then multiplying by 1,000. So that's what that per mil value is, okay? So nothing to be worried about. And you can see these numbers here, they're pretty big, and so basically when they are getting closer to 0, this is indicating more saline conditions in the lake. The other thing I want to talk about is this ratio of sulfur to calcium. So we measure all the elements down these cores, and what this is telling us about are changes in the minerals in the sediments. So we have calcium carbonate, we also have gypsum. Gypsum is an evaporite mineral. It forms when water is evaporating. So when you have really salty water, as water is evaporating, you should also get gypsum. And these two actually do kind of co-vary quite nicely, as you'll see. I'm not going to spend time talking about the other 2 tonight, but if you are really interested in the Q&A, I'm happy to run you through those. Okay, so where's the climate information now? So we've converted delta D into salinity of the lake in parts per thousand. This is actually, you could go to the lake and measure with a handheld conductivity meter today. And in fact, we've done that. That's where the stars are located in this subplot of the modern sediments. And so usually when we go to this lake, as I mentioned before, it's brackish to fresh. Brackish means it's not as salty as seawater, but it's not totally fresh. Okay, it's usually about 20 parts per thousand or so. The reconstruction of salinity from the lipid isotopes suggests that salinity does get higher in the recent decades. This is sort of a decadal time scale here. But then you look at the older part of the record, this line here is basically the hiatus, right? We don't have any sediment. If you look at the deeper past, you see that there were decades, maybe even a century plus, where salinity was very, very high. And this was also a time period where we got gypsum precipitation, and we do not get gypsum forming in this lake today. It's not salty enough. Okay, it's not evaporated enough. And so, what does a really salty lake mean? Well, it basically, from what I was talking about with the monitoring, the groundwater lake is going to get more saline as there's reduced precipitation and increased evaporation. Okay, so we're recording a pretty prolonged dry period here, and in these periods as well, but not as extreme. Okay. So now let's dig in and start to test those hypotheses that I talked about earlier. What can we do with this record and the Washington Island record, now putting them together? So when we do that, we see some interesting things emerge. So Christmas Island, our salinity record is in yellow, Washington Island is in red. The very cool thing here is that on Washington Island, the same variable was measured in the core, the delta D of lipid extracts, delta D values of lipid extracts, and it was converted into salinity in the same way. So apples to apples here. So today, in modern times, Washington Island's lake is fresh, which is what this red dot is supposed to represent. And, Christmas Island is brackish. It can be quite fresh. It can get a little bit above ocean salinity, but nothing like the 1000 CE. So first take home point is that overall in both these lakes, it's suggesting that in the last millennium, things were much more saline in these lakes. And that implies that there was reduced periods of reduced precipitation during prolonged periods. Okay, so that's the first interesting thing. Your eye is probably naturally drawn to this prolonged period of high salinity on Christmas Island, in the Christmas Island record. And so this is really interesting. And it's very cool because it also coincides with the migration of Polynesians across the Pacific Basin. And so some archaeological papers have suggested that a change in climate at the time was somehow connected to that movement and the occupation of islands in the Eastern Pacific. So that's pretty cool. But what does it mean climatically if it was really, really dry on Christmas Island during this time? That would support, a stronger Pacific walker circulation. It could also mean that rain band, it's north today of the island, but maybe it was even further north. That's also a possibility. But it seems likely that, to me at least, that Pacific walker circulation was very robust at this time, and it was just a very dry period. Unfortunately, the Washington Island record is too short and does not overlap this time period though. And then here, this is where Christmas Island's lakes, it dries up. It's not there. We don't have any data, unfortunately. But here, Washington Island shows this really abrupt increase in salinity, suggesting that it got very dry over this island. Okay. And so basically, if we're interpreting the absence of the lake record as, dry conditions, it doesn't necessarily support, the rain band moving south over Christmas Island. So if that rain band had moved south, Washington Island, which sits under it today, would have gotten drier, but Christmas Island would have gotten much wetter. And we don't seem to see much evidence of that. In fact, they both seem to be dry at the same time, which again is more evidence pointing towards that stronger walker circulation. Okay, so I'm into the final scientific vignette for everyone tonight. So tune back in if I lost you. We're going to talk now a bit about geobiology. So we can learn a lot about climate from these lakes, but they also house these remarkable microbial mats that may have something to tell us about carbonate formation and various nitrogen and carbon cycle processes. So what are microbial mats? They look something like this, so they can be very diverse. They're essentially these layered structures with very distinct bandings. And each of these, colored areas, these layers, has a distinct community of bacteria and archaea. And they also have very distinct geochemical and physical properties. So, pH and alkalinity will be very different from layer to layer. Light availability is going to drop off below that first layer very quickly. And so you're going to change, the environment, the microenvironment, and that's going to change what types of microbes can live in that environment. And so we are fascinated as earth scientists by microbial mats because the fossil counterparts of these mats are some of the earliest evidence for life on Earth. And we think that these mats in early Earth played a key role in putting oxygen in the Earth's atmosphere through photosynthesis. Okay, so that's why people study microbial mats. And so what are we going to do with them? So Christmas Island, apart from being a great place to study tropical Pacific climate, can also be thought of as kind of a natural carbonate laboratory. And so we are certainly not the first people to look at the microbial mats of Christmas Island. And people have investigated in other papers a wide variety of topics, how do these mats form carbonate minerals? And just, who's in these mats? What's the taxonomy of these different layers? Questions like that. Okay, so this is established in the literature. Our approach to this, though, was a little bit different because, we're paleoclimatologists, we're geoscientists, And, we already have this very broad transect of sediment and water samples from a variety of different lakes. And I will say most of the work that's been done on Christmas Island has been done on, with microbial mats, is done on maybe one lake or two lakes. No one's ever looked at many lakes together. And so we wanted to get a sense of the relationship between salinity and the microbial communities and the different types of carbonate minerals across a wide variety of Christmas Island lakes. And then, we also wanted to get a handle on, what were the microbes, what are they actually doing in the lake sediments? What functional genes are present? And can that shed any light on some of these pretty common measurements we make as paleolimnologists to understand past changes in both climate and the environment? So things like nitrogen and carbon cycle changes. And so, the hypersaline lakes and the brackish lakes, remember Lake 30 was a brackish lake, they look different. The color of the microbial mats is different and the mineralogy is different. So our hypersaline lakes, where we have, high salinities up to 140 parts per thousand, those generally precipitate aragonite, which is one form of calcium carbonate, whereas the brackish lakes precipitate more calcite. And 16S rRNA gene sequencing showed us that a lot of the same phyla, bacteria, and archaea are the same in both lakes, but there are some differences. Like we see this guy Firmicutes, more abundant in hypersaline lakes, and this is this phyla. They can osmotically regulate their cytoplasm, so they can survive in hypersaline environments. Whereas we find more cyanobacteria in brackish lakes, and it's well known that cyanobacteria don't like it when it's very salty. It's an abiotic stressor, okay? So, that kind of answered the question of who's there, but what you really want to know is what they're doing. I will just add a quick aside though. One interesting thing we want to do is that all of those measurements we did were from samples from 2014, but all these lakes have totally changed after that big El Nino event. And so we have a fresh batch of samples from these same lakes taken in 2019. And so we want to compare the microbial communities across this event and see how dramatically it changed who was living in the lake. So that's something that's ongoing. But, going back to that question, one thing we really want to know is, what are these microbes doing in the sediment and how does that influence the things that we measure in our sediment cores and try to interpret as changes in past environment, past climate. So my student Ming Fei is working on this problem with a lake core from Lake 1, which is close to Lake 30. This is a lake that can be a bit more saline, or it's more towards the hypersaline end of the spectrum compared to Lake 30, but it largely looks very similar compared to the Lake 30 sediment, as you can see. And so he's doing this cool combination of developing metagenomic libraries from different sections of his lake sediment core. And he's combining that with sort of traditional methods that we use all the time in paleolimnology. So, describing the sediment, the mineralogy, dating the sediment, measuring the geochemistry. And so basically what he's doing is he is sequencing all of the DNA in a smidgen of sediment from each of 5 different depth intervals. And with that, you can see who's there. You can look at taxonomy like we did earlier, but you can also look at the functional genes that are present. And so he did this first off looking at the nitrogen cycle, because when we measured the stable isotopes of nitrogen, this is another isotope system that you can measure, he found a really big change. And so this is a big change for a lake system. And so this variable, we're looking at the nitrogen 15 relative to the more common nitrogen 14. It's measured all the time in lake sediment studies. It's interpreted in a variety of ways. It often is related to productivity. It can be related to things like atmospheric nitrogen deposition with pollution, eutrophication from pollution, all these sorts of things. But the thing about this measurement, this variable, is that it's very wishy-washy. It's really complicated, and there's a lot of uncertainty. Okay, but so what we were hoping to do is that maybe if we see what nitrogen cycle genes are present when we have high delta N15 values and low delta N15 values, it might shed some light on why we see these numbers that we're seeing, these per mil values. And so what you're looking at here is a heat map of different nitrogen cycle genes that Ming Fei identified in his 5 sediment sections from this core. this is the surface sediment from 0 to 5 centimeters, and this is the deepest part of that core. Okay, so a darker blue means that gene is more abundant in that sample. And so we found some interesting things, and I know we're getting towards the end of the talk, so we're not going to do a deep dive into this, but he found some interesting things. So he suggested that, even though in these surface sediments we get a lot of sunlight, we have a lot of N fixation genes, we see a lot of cyanobacteria that would suggest that N fixation is really important. We have these really high delta N15 values, which doesn't make sense with a lot of N fixation because that has a very low delta N15 value. So this exercise showed that there are a lot of genes related to denitrification, which is a process that enriches or makes the delta N15 values of the organic matter that we're measuring much higher. So that was pretty cool to see. And then to explain the really low values deeper in the core, there's not a lot going on at depth, right? It's a low energy environment. But he did see a lot of one of these N-oxide reduction genes, and this produces ammonium. And this actually has a low delta N15 value. And so we could just be measuring this along with the bulk organic matter, and that could be what's driving our values lower. And the take home there is that when you're measuring the delta N15 value of bulk organic matter, deeper in the core, it might not be preserving a record of what was happening at the surface. Okay, so I'm going to wrap it up now, and I think that leaves us a lot of time for questions. So 3 points to come away with. Hopefully I've shown you that the lakes of Christmas Island, many features of them are strongly controlled by moisture balance changes in the atmosphere, precipitation and evaporation. And we can use that to understand past changes in moisture balance in the past. So what we're seeing from our sediment cores is that there were periods that were much, much drier in the last millennium relative to today. And that enables us to test some of these hypotheses about what the tropical Pacific was doing during the last millennium. And we don't think there's evidence of an ITZC shift between Washington Island and Christmas Island. I think evidence is pointing towards, at least in the central Pacific, a stronger Walker circulation at that time. And then finally, combining these traditional kind of stable isotope measurements that I've been doing in my field for decades now with more recent metagenomic techniques is helping us understand our lake sediment records much, much more. So I'm going to stop sharing. I will leave it at that. And I am here to take your questions.
Okay, we have a couple that have been posted in the chat, so I'm going to read them off so that they go into the record very well. My colleague Ricardo Covarrubias asks, how are these weather patterns correlated with solar activity?
That's a good question. So what I think of immediately is that there's not a strong relationship. The latest work that's been done looking at those external forcings on tropical Pacific climate, it shows that volcanic forcing seems to have a pretty big effect. It's one of my colleagues' work. So, but I don't think there's a really compelling signature of solar radiation or changes in solar forcing over the last millennium, at least.
Okay. And I think for our audience, maybe clarify what you mean by forcing.
So yeah, okay. So when I say the word forcing, it's sort of just like you change something and then that influences the climate system. So we're changing how much heat is coming into the climate system, or with volcanism, you are putting a lot of sulfate aerosols high up in the stratosphere during a big volcanic eruption, and that can block incoming solar radiation and change the climate. So yeah, that's what I mean by that, just changing the amount of the radiative balance.
Great, thank you for taking the time to do that. Here we go with some more questions here. What is the cause for a big El Nino event? Does it correlate with something?
Yeah, hard question. That is a great question. There's not a clear answer, and I wish there was. A lot of these big events, they do occur when we have these westerly wind bursts. So sometimes there's just these anomalous winds that come out of the Indian Ocean. They start over by the coast of Africa, they blow across the Indian Ocean, they just keep kind of propagating across the Pacific, and that kind of pushes all that warm water to the east. And so yeah, the westerly wind burst would be pretty critical. Yeah, it's a good question.
Yeah, it's like asking why we see certain solar cycles where there's so much more sunspot activity and so on. So yeah, I don't have a great answer myself. All right, next one here. Have other islands or chains of islands been studied in the same manner regarding paleoclimatology? If not, can you speculate as to why not?
Yes, other islands have been studied. So, I just talked about Washington. We had a short time together tonight, but there's a lot of work on other islands. So I've worked in the Galapagos. So there's a lot of lake sediment records developed from the Galapagos, a lot of coral records from the Galapagos, a lot of coral records from the Line Islands. And then when you go into, more of the Western Pacific or the South Pacific, there's many more islands, and a lot more has been done with lakes, with cave deposits, with corals. Yes, and so that's part of the problem. Everyone is developing these paleoclimate records. They make their record and they don't all match up beautifully. And so this is where the controversy comes in with these competing hypotheses, in fact.
Okay, and then one from Jeff Bryant, who's actually going to be our speaker for next month's Kaler lecture. He says that I've seen studies of Delta 018 for ancient paleoclimate, a recent one studying carbonate muds, I think dating back to the Cambrian period. That goes back far. How far back can these go? And why is Delta 018 a good measure for temperature?
So good question. So yes, so a lot of the time, Delta 018 is used as a paleothermometer. That's because the partitioning or fractionation of the light isotope from the heavy isotope when something like carbonate rock is forming is temperature dependent. So it's a thermodynamic process. And so we can back calculate out temperature from that. And so this is actually the first way they were really stable isotopes of oxygen were really used for paleoclimate. And then in a place like the tropical Pacific, we realized that they also track the water cycle really nicely. And that signal is much louder than the temperature signal because the temperature changes are small in the tropics, but the rainfall changes are huge. And, carbonate itself, it's all a question of diagenesis. So is that original material preserved, from hundreds of millions of years ago? If so, then yeah, you can measure those Delta 18 values and infer past changes in climate from them. But you just have to make sure that you haven't had like recrystallization or something like that.
Wow. All right. Then there's a question that got thrown into the Q&A. Why do La Nina and El Nino happen? And why are they named the way they are?
Yeah. So, it's this phrase internal variability, so something can happen to trigger an El Nino event, but it's not forced like with the volcanoes and the solar radiation that we were just talking about. It's just the natural oscillation in the climate system, and they're named that because this phenomenon of warming ocean temperatures during an El Nino was first observed in the eastern Pacific off the coast of South America around Christmas time. And so El Nino means the Christ child. So it's just referring to the time in which these really anomalously warm waters would appear, and those warm waters would bring with them to this normally cold region, tropical fishes and things like that.
Very cool. All right, now we've got another one here that is asking for your comments here. Let me go ahead and I guess I'll read through all of Mark's comment. Humans are pouring greenhouse gases into the atmosphere at an unprecedented rate. Given the inertia and lethargy to date of our American government in this area, I assume that it will proceed apace for some time to come. I know that it's not really your area, but would you care to make any predictions about what climate in central Illinois will be like in 50 years?
So I don't think about Midwestern climate projections as much as I think about the tropical Pacific, but temperatures are projected to increase. They're not going to increase as much as temperatures will rise in, say, the Arctic, where things are warming up much faster, but they are going to rise. Precipitation is the big thing, though. This is a part of the world that's projected to get wetter. And then it's a question of how does that increase in precipitation manifest itself? Is it just a kind of an increase, a bump up in the average, or are there more extreme rainfall events? And things are suggesting that there might be more extreme rainfall events.
Yeah.
But yeah, that's an active area of research, so it's not set in stone, but yes, that's what people are thinking.
All right. Okay. Jeff has a follow-up with what he was talking about. In the water, not in an inland pool, would DO18 track precipitation as well? For example, shallow seas, deep water, etc. How would the rainfall cycle modify the DO18 cycle and swamp the temperature effect?
Yeah, so the temperature effect is about 0.2 per mil per degree Celsius. So it all depends on whatever you're measuring, what is the range of variability that you see. Imagine in a lake, since that's what we've been talking about, or shallow sea. Okay, so you have a lot of rainfall. The delta O18 value of rain is more negative, so it has fewer of those heavy isotopes. And so that rain, that freshening, brings all of this isotopically light water into that surface water reservoir. And so that's going to make that surface water reservoir, be it a lake or a shallow sea, more negative. Okay, and then if you have less precipitation, you have evaporation of that shallow sea perhaps, or that lake. Evaporation preferentially leaves the heavy isotopes behind and it takes up the light isotopes. And so that residual pool of water that's being evaporated is going to become more and more isotopically enriched in the heavy isotope over time. So the delta 18 value of that pool of water that's evaporating is going to go up. And that can be, per mil changes. So instead of 0.2 per mil, like several per mil changes. So that's what I meant by swamping the signal, if that makes sense.
Very cool. All right. I don't see any other questions, so I'm going to throw in one of my questions, but I don't think this has much to do with your talk, so I apologize in advance. Do you feel that that term little ice age is still appropriate based on what we now understand about the temperature record from that part of the millennium, the last one?
Yeah, those periods are almost problematic, I would say, because
yeah.
I mean, they're not globally uniform, and so what do they really mean? But yeah, I think it's probably sticking around.
Okay. I had heard one time that maybe that little ice age idea was just based off of what was experienced in Europe as opposed to what was experienced globally. Yes. Yeah. Okay. Good. So I'm glad I remembered that correctly. Thank you. All right. We have another question here from Heidi. How did you know you wanted to study lake sediments?
Yeah. Well, this all came about because I had a research experience as an undergraduate, and I got to go to Western Ireland and core lakes and bogs as part of my senior thesis and part of this summer research experience. And the professors I was working with were really interested in this this thing called the Younger Dryas, which maybe some of you have heard about. And so the Younger Dryas was this abrupt period of climate change after kind of at the end as we were coming out of the last glacial cycle. So the last glacial maximum, things were warming up. And then boom, over a period of decades, things basically went back to glacial conditions. So it was this really abrupt change that would have happened over the lifespan of a human. And so we were looking for that in the lake sediments. And so that wound up being my senior thesis project, and it got me hooked on lake sediments.
Very cool. All right. Okay, we got another question here from Chris. Due to the history of atomic detonation near or above the island, are there any special safety precautions that you need to consider when you're out in the field?
No. We have never brought a Geiger counter with us, but there doesn't seem to be, like, it's not actively like irradiated, so it's not emanating radiation or anything as far as we can tell. I mean, the islanders still live there without precautions. I mean, I don't know if anyone's been there with a Geiger counter and what's going on, but it seems to be safe. One thing we have done is we've looked for radioactive isotopes in our lake sediments to measure them, and we thought we would see them. It's like, oh my gosh, this is going to be a very clear marker. We will see a big peak in cesium-137 due to these bomb tests, and we never found it. So what we're thinking happened is that these bombs were detonated in the atmosphere, a kilometer or so above the surface. There are strong trade winds, right? They're blowing from east to west, as I was talking about. And so that all of that radiation, or a lot of it, might have gotten pushed away from the island. And so it might have settled out over the ocean to the west. I mean, that's one hypothesis. But then again, a lot of people got cancer. There's a lot of evidence of birth defects and things in the population. So that doesn't totally pan out, I don't think, so it's tough. I don't know. Yeah.
That's interesting. Yeah, is that island downwind from a few of the French or American tests from the Cold War?
Is there an island downwind?
Well, I mean, is Christmas Island downwind from some of the past tests that were done in French Polynesia or from Micronesia, which is where the Americans were testing and stuff, so.
It's really far away. Yeah, like from
Yeah.
the pretty far away.
Okay. Well, you'd mentioned that there were some birth defects. I was like, oh, it's sad to hear. All right. Let's see here. Mark asks, did you find the evidence that you were looking for in Western Ireland?
I think, yeah, eventually when the papers were published, they did see, they had a Holocene records the last 10,000 years. And I think they did find that Younger Dryas. I mean, I think my dissertation, or my thesis wound up being about just the Holocene. So like 10,000 years ago to present day, so.
Okay. All right. And then Jeff has a question about the Younger Dryas. I believe that recent research supports a meteor impact may have triggered the Younger Dryas, perhaps the recently discovered large crater under the Greenland ice sheet. Any updates on the causes for the Younger Dryas?
This is something that people get very angry about.
Oh, good.
It's really interesting, but I think people are moving away from this impact hypothesis right now until there's more evidence.
Yeah.
Okay. But yeah, I did just read about the crater under Greenland, the Greenland ice sheet. That'll be interesting.
Yeah,
once the ice melts to see.
That'll be cool to see. Right. Oh, let's see. We got another question in the Q&A. This is kind of related to the last question about why you chose what you did to study. But if you didn't go into the specific area, what field would you go into instead and why?
Yeah, I think about that a lot. I've often gone, I've always liked anthropology. I took almost enough classes to be an anthropology minor in undergrad, and I feel like I could have gone in that direction pretty easily. But who knows? Maybe I should have just gone to med school. I don't know. I don't know. Yeah.
Gotta imagine you're doing pretty well for yourself.
I guess I will say, if I had it to do over again, I think, in terms of like looking towards the future, what's the most important thing that we could do is maybe engineer a way out of the climate crisis by actually figuring out how to take CO2 out of the atmosphere at scale or something like that, some massive problem that needs to be solved.
Yeah, there was a pretty amazing NOVA episode in the fall about can we cool the planet? And they talk about all those technologies. Okay, let's see here. Jeff's following up on Younger Dryas here. What are the leading explanations right now? Is it freshwater disrupting salinity rivers in the ocean?
Yes, it is. So we talk about there were these large lakes that formed at the margin of the retreating Laurentide ice sheet as it was melting back after the last glacial maximum. They were big lakes, and they would catastrophically flood. They would just dump all of their water through channels out, and eventually all that water would wind up in the ocean, either through the Mississippi or through the St. Lawrence Seaway or a few other rivers. So the prevailing hypothesis is still that there was a big meltwater flood, and all of this fresh water got dumped into the North Atlantic Ocean. And it created kind of a freshwater cap because it was less dense than the salty water. And so it sat there at the surface, and it disrupted ocean circulation. It disrupted the important bringing north of heat, which keeps things warmer and more mild in those higher latitudes.
Sounds like it would have caused a little ice age.
Yeah.
All right. Kelly has asked, do you have any advice for a student who might want to go into this field?
I would say try to get a research experience as an undergraduate, like a real bona fide research experience that culminates in writing something like a thesis, because this whole process, a lot of it winds up, less of it is lab work. I mean, that's a big chunk of it, measuring things, going to the field, collecting materials, measuring them, but then figuring out what they mean, writing, making figures, that's what takes up the most time. And so you'd want to get experience doing that.
Yeah, sounds like it goes back to your, was that an undergraduate experience in Western Ireland? Is that what that was?
Yeah.
Excellent. Yeah. And then Heidi is asking about the certificates behind you. So you might have to move your head and explain what you have up there on the wall next to your map.
My undergraduate degree from the College of Worcester.
Mm-hmm.
That's my BA in geology. And then I was in Phi Beta Kappa. And so that's my Phi Beta Kappa certificate.
Very cool.
Which I carried around with me for decades as I was getting my higher degrees. And then once I finally settled here in Illinois, I got them framed and finally could hang them up.
Oh, wonderful.
Oh.
All
College
right.
of Worcester, huh?
Yeah.
Okay, is that where you're from?
I'm from Western New York, actually. So
Okay.
Buffalo. Are you from Ohio?
I am very much so. And our science Olympics used to be at the College of Worcester. So we would, it was quite a drive, but it was just such a wonderful campus. I loved it.
Nice campus.
Cool. All right. Well, anyways, we ran long, but we had lots of great questions, and I
So
think
worth
we had a great time hearing from you. This was so informative and so interesting. So, Dr. Conroy, thank you so much for taking the time to present at the James B. Kaler Science Lecture Series this month. And it looks like our audience really had a good time with this. Before I move on to our next show, I want to mention that our next lecture is going to be given by one of the people in our audience, Jeffrey Bryant, who works for Wolfram Research. He is going to give a talk about the birds that you can find in East Central Illinois. He's quite an avid birder, and he's got lots of amazing photographs that you can actually find on his personal Flickr page if you just go around and look for it. But he's going to show a few of them here, and I think it's going to be a really pretty lecture at the very least. We hope to get him again to give a talk so we can put some of his photographs up on the dome when we can do this in person too.
Oh.
So yeah, that will be 4 weeks from today, which I believe, because it's February, is going to be February 5th. I'm sorry, March 5th. That's what I should say.
Yeah.
So we'll have that. Now, for those of you who'd like to stick around, our next show is going to be Prairie Sky Spotlight on Mars Missions. Wielene is actually in the dome, and she is going to be presenting that one, but I will stick around to moderate. Dr. Conroy, you're welcome to stick around as much as you like, but you have done great work for us already, so thank you so much. It was a pleasure to meet you.
Around. I have to put my baby to bed though, so I'm
totally understand.